point mutations can have catastrophic effects on the proteins that mRNA sequences are supposed to synthesize. If you look at codon 3, you'll notice that this codon is comprised of adenine, uracil, uracil. If we were to insert cytosine into right there, this codon would have changed to adenine, cytosine, uracil, which codes for a completely different amino acid, changing the protein. And you'll notice that as it was inserted, all the rest of the strand changed as well, shifted over by 1. So these new codons code for completely different amino acids, ruining everything. Likewise, if we removed it, everything shifts by 1. These are all different amino acids from what they were previously. In the case of this protein and its mutant, in order to figure out what the mRNA sequence is, we're going to have to cross-reference them with each other by looking at the codons that would code for them. So methionine and tryptophan, both of these are just coded by 1 codon each, but we'll notice that a problem arises at codon 3. Phenylalanine and isoleucine, definitely not the same thing. They're coded by completely different codons, and we're going to write those down, as well as the codons for the rest of the protein. And we have to remember to write the stop codon. If histidine is the last amino acid in this protein, there has to be a stop codon. Let's write that down too. And when you're doing this, you have to look for patterns. Notice that there's a UUC right here. And just one over, there's a UUC with the C from this normal protein being in the next codon for this mutant protein. That UUC there must correspond, and this brings our attention to this rogue adenine. There's no adenine anywhere near where it's located in the mutant protein instead normal protein. So we're just going to say that is most certainly an insertion, and it's moved everything down. If we correspond each base with the base in the normal, we'll be able to figure out what the actual order is instead of having to guess every base or so. And once we do that, with the exception of these at the end that we can't cross-reference with the mutant, we'll find these orders right here for both the mutant and the normal. And as you'll see, it's just an adenine that came in and pushed the rest down, ruining this protein, making it completely non-functional.